Hello again. Thanks for the organizer to let me talk today about The Sky is the Limit, a cloud-based proteomics platform for the masses. This morning, I had my science wardrobe on, and over lunch, I decided to change into the startup wardrobe, partly because I spilled some food on my trousers, but now I have the time to talk a little bit about where I'm coming from and what MS8, the company that sponsored ISMB, is actually doing, and the product that I'm describing here is part of that. So MS8 is a company that transforms proteomics with AI, and transformers are actually a big part of it. So we love transformers and natural language processing and their applications to proteomics data analysis. And in this talk here, I'm not going to talk about transformers, even though I love them. I'm going to talk about something that we have realized that is actually a bottleneck in the entire workflow, going from an idea, a biological sample, all the way to the biological conclusions in the end. For those of you that regularly work with mass spectrometry data, and I suppose that's all of you, you know that instruments, MALDI-TOF instruments, but also other instruments got better over the years. They are more sensitive. There's a lot of automation built in. They're very usable, and they're becoming more easy to be applied even by non-specialists. You need less experience to generate the data. However, we have identified that the workflow from data all the way to conclusions can be very manual. So one thing that I experienced myself when I was in a lab was that we generated the data on a beautiful instrument, and somebody had to go regularly to that instrument and pull that data off the acquisition computer, because the hard drive of the acquisition computer would fill up. And if it fills up, then the machine is down and everyone cries in the lab. So that was kind of manual. So we went there with a hard drive and we pulled the data to a hard drive and then carried that hard drive. Please don't trip, data worth hundreds of euros, thousands of euros would be destroyed in an instant, on an external hard drive to a processing PC, store it in some home directory, and then sort of, analyze it there, and then store it in a completely random folder someplace. And then once it's time to write up the paper, you've got to go back through lists of folders and ask yourself, God damn it, where is the one experiment that I've done in the past? I don't remember where it is. So the companies that sell mass spectrometers nowadays do try to help you with that process. So there's some automation built in. But the bioinformatics that are associated with analyzing the data, we feel are very unstandardized. Result storage is unorganized. I mentioned this. And once you actually then go and want to ask your data some questions, either using R, Python, Excel, looking at the data, it becomes very sort of non-streamlined, and it depends on the skill set of the researcher all of a sudden how much you can actually get out of the data. So we think that this is actually a key problem that needs solving, and at MS8 we tried to tackle that problem and essentially develop something very akin to what Matthew this morning from AstraZeneca was presenting, and that is essentially a platform for the analysis of proteomics data from the instrument all the way to data interpretation. Don't mind the very boring name. We couldn't come up with something better. We went through several iterations of AI-generated names, but MS8 platform was the best thing we could think about. But given the fact that MS8 stands for Aiding Mass Spectrometry with AI, it has some ring to it still, so we stuck with it. It's probably okay. So what are the key features of that platform? Well, it's an all-in-one cloud platform that is accessible in your web browser, either on your computer or from your phone. So if you're that type of person that wants to monitor progress of your searches from your phone while you're sitting and watching Netflix, that can help you with that. The features in detail are that it seamlessly scales with data volume, and that is very important in a day and age where mass spectrometers all of a sudden generate a factor 10 more data per time than they did like 5 years ago. So when you think about, say, orbit traps, for example, they are great instruments, very high accuracy, very beautiful data comes out of them. However, they are slow. So people used to move through TOF type instruments and are generating 10 times the number of spectra in the same amount of time. I don't want to be a data scientist or an engineer at the EBI taking care of pride, because once that data systematically hits repositories, they are going to have a bad time. This platform here, similarly to the EBI, of course, scales with data volume. So the more data you generate, the bigger your repository can get. And of course, that is only achievable if you're going away from sort of your local data center and deposit the data in the cloud instead. With using the cloud, of course, comes the advantage of having access to elastic parallel compute. So if you have hundreds or thousands of raw files, you can analyze them in the same time as you can single raw files. And that is a big advantage if you're very time restrictive. You need your data yesterday because your boss asked you to, hand you that presentation for that famous conference that you need to go to. The results storage is also baked in. So while we store raw data, we also store the results, and the results are stored in a, for the lack of a better word, another buzzword here, a data lake. It's effectively deposited on S3, similar to what Matthew has been showing to you this morning, and that allows you to be very flexible and very space and cost efficient with result storage. We allow interrogation of the data through various different means, and I will show a couple of them in a second. But the best feature I think that is coming is that there's API access, so you can effectively directly interact with the data lake through an API and analyze the data in a virtual machine on AWS as well. But that API can not only be used to interrogate the data, but also to actually start searches. So if you want to start a search with a very specific search parameter, you can actually do that. If you want to monitor the contents of a specific folder on your, say, acquisition computer and upload that data to the cloud and then get rid of it so that your acquisition computer doesn't fill up, you can do that too. And it comes with a subscription-based pricing model. So effectively, what we have designed is a front end and a back end, and the back end essentially stores the data, handles all the compute, and ingress to that back end is either via the CLI client that I mentioned earlier, or via the UI, or via typical FTP Explorer tools that you know and love. And the backend here is perfectly capable of dealing with multi-gigabyte uploads. But what I would advise against, and I think what the field sort of realized that is not sustainable, is don't acquire your entire experiment, your thousands of raw files or hundreds of raw files, and then ask the question, how do I analyze that data? Ideally, you would acquire a raw file, upload it already, and then acquire the next raw file, upload it, because then you are not tasked with uploading terabytes of data to the cloud and then needing to wait for that upload to finish. That's a poor experience, in my opinion. Right. We heard that this morning already that metadata is actually quite important, and it gives context to your experiments. So in my opinion, there's two different options to deal with metadata. This morning, we heard a very structured way of capturing metadata with controlled vocabularies and graphs of decisions that are attached to an experiment. And that is great if your experiments fit to that type of controlled vocabulary and fit to that type of experiment, and you do very standardized experiments over and over again. But we felt that this is probably not what everyone's going to do. So instead, we handle metadata on the platform slightly differently, and we do that in a Google Mail type of fashion. So every raw file, as well as every search result, you can tag with arbitrary free text tags. So you can add tags, for example, here with specific sample identifiers, with specific modifications that you searched, instruments, maybe labeling methods, specific publications that are associated with that. These tags, these metadata tags, allow you then later on to find your raw files back, as well as use them as contrasts in your statistical testing. So later on, if you have a search of multiple raw files that belong to a specific project, you can identify these raw files by project ID, generate a search from them, and then attached to the separate raw files, you might have cell line names and replicate names, and then you can separate your replicates and contrasts by that and do t-tests, clustering, etc., based on these tags. It is not implemented yet, but what we envision doing is automatically tagging the data based on some information that is contained in either the searches or the raw data. So for example, extracting the analyzer, extracting whether it's a DIA or DDA type of experiment, all of that can be automated and could be automatically tagged and found later on in your list of raw files as well. We envision also that we can integrate this system with LIMS systems in the future. So if you're a developer of a LIMS system and you have an API available and you want to pull in the data that is stored in the platform, in our platform, that is certainly possible. And the same thing is possible if from the LIMS system you say, well, you have here a couple samples and here's the raw data stored in the LIMS system. Please submit a search for that raw data on the MSA platform. That's also possible since it's all API controlled. The tags, once you add a tag, it's persisted in the database. So if you want to reuse the same tag, you will be prompted with a list of tags that you have previously created. So it kind of serves as a halfway in between controlled vocabulary and fully free text tag system. So if you have, for example, HeLa cells that you frequently analyze for QC purposes, for example, then we show past tags that you've given in order to not generate duplicates, for example. The MSA platform will be powered by our flagship product, which is CompMS. CompMS is a search algorithm, for those of you that don't know, that can deal with DDA, DIA, and PRM data. Currently, it is integrated in Proteome Discoverer, but with the MSA platform, it is now available without Proteome Discoverer. So there's no need anymore for Windows environment. It's all Linux-based, it's all API controlled, and I know many of you sort of dislike the Proteome Discoverer interface. Here is a solution that implements all the functionality available in PD in combination with CompMS in a complete web-based environment. It is, however, designed as a modular system. The platform is designed as a modular system. What does that mean? It means we are not restricted to CompMS. I think that's a pretty cool concept. We could plug in any search engine here and make it available for use with the platform. The only thing that would be required from our end to do is to write the adapters, input and output, and then effectively merge the data into our data format. So if you're a developer of a search engine and want to sort of have it scale out to thousands of machines in the cloud, then please approach us. We are open for collaborations. And of course, if you are integrated in the platform, that would result in a kickback to you when searches are done with your search algorithm. Why would you be interested in actually collaborating with us on that? Well, I mentioned it earlier. We have a fully scalable Kubernetes cluster in the back on AWS. So you can very fast parallelize your searches over your different raw files. Here I've brought a graph of a number of raw files and how long it took to process them. On the x-axis, you have the number of raw files, and y-axis is processing time. And you see that there's no real difference between processing 1 file or 100 files. And the increase in computation time is negligible. So I think it's a good opportunity to try and take advantage of a structure like this if you're a developer of a search engine. When I speak of results, all the results are effectively organized in an experiment-centric fashion. Every experiment consists of many raw files, but experiments can also consist of single raw files only. And I think that's the big advantage of the solution that we have here. The fact that you can search individual raw files and then later on actually combine them into bigger experiments is a very important feature, in my opinion, because what it allows you to do is search every raw file individually. So do QC on your experiment as you measure the data. And then once you finish the data, all you have to do is combine these experiments into one big experiment, and we then don't repeat all the searches again. You don't have to research the data. The only thing you have to do is calculate peptide FDR, protein grouping, protein FDR, Quan, and that takes no time at all. So by the time you've acquired the last sample and analyzed the last raw file, you are just a couple of calculations away from finishing the entire project. And what Matthew brought up this morning, that, assume that, say, you're looking at a patient study and a patient withdraws consent, you have to delete that raw file and maybe even the entire data behind it from the platform. You're able to do that. But then reanalyzing that data doesn't need you to research it. So again, you can just delete the raw file, delete the results associated with it, and combine all other raw files but the one that you wanted to exclude, and you still have consistent data in a matter of a couple of minutes. You can explore the results directly in the browser. So you can ask questions like, how many proteins have I identified? Are the number of IDs okay in comparison to the gradient length that I've been using? Is my favorite protein there? So here, for example, we looked for a particular protein that we are interested in, and we can see that it is there. And that is, of course, one way to look at proteomics data. But as you know, tabular data and looking at tables in the browser is just, not really exciting. So what we also built is a visualization dashboard, and that will be growing in the future. We have a couple of basic visualizations already present, and you can effectively use the tags that I mentioned earlier in order to generate contrasts and perform volcano plots and scatter plots and the like. And those dashboards are easily shareable then with your colleagues as well, in order to foster collaboration and interaction. So yeah, all that is left to say is please try it out. It is available under platform.ms8.io. Please let me know what you like, what you dislike. And we will gradually admit people to the platform. Once you register, we get an email, and then we get back to you once you're activated. So with that, all that is left to say is thank you very much for your attention. And if you have any questions, feel free to approach me.